Hi everyone, my name is Paula Escuer and I'm a PhD student of the University of Barcelona under the supervision of Julio Rozas and Alejandro Sánchez Gracia. And today I'm going to speak about the chromosome level assembly of Canary Island endemic spider Dysdera silvatica. So first of all, I would like to start my presentation speaking about the first steps of the evolutionary studies. As you all will know, the man of the picture is Charles Darwin, who introduced the key concept of natural selection. Here at the right picture, we have his famous sketch of the Darwin tree of life, which represents the evolutionary relationships between species. So Darwin made a turning point in the evolutionary theory. However, this field has been extensively developing during the last century and even nowadays. So in order to make our contribution, we first need to choose a biological model. And we choose the spiders of the genus Dysdera. Dysdera's spiders are from the order Araneae and the family Dysderidae, distributed across Europe, North Africa, Central Asia, and North and South America. These spiders are also active nocturnal hunters and during the day are sleeping in their cocoons, under stones, dead logs, leaf, or even some of them live in caves. So these spiders, some of them have a particular interesting thing that some of them have been specialized in eating wood louse. As you can see in the picture of the middle, or more or less, because the wood louse is very well mimetized with the environment, there is a Dysdera spider eating one wood louse. So why are these spiders interesting from an evolutionary point of view? So these spiders have an adaptive radiation in Canary Island. From 250 species, approximately 50 of them are endemic from Canaries. And this means that from one or several ancestral forms, there was a rapid diversification, thing that we can observe here in the phylogenetic tree. So the main theoretical basis of my PhD is basically to identify the genes and genetic elements to understand this radiation and focusing in the trophic specialization of these data. These dietary specializations have been supported by morphological and metabolic evidences. For example, in the specialist spiders, their chelicera are modified to better catch and break the defenses of the woodlouse, and there are also evidences that specialist spiders digest and assimilate much better this kind of prey than another, on the contrary than generalist spiders. So we select as a model to test our hypothesis that we select 5 species of Dysdera, 2 pairs that were phylogenetically close, one generalist and one specialist, and one outgroup that is Dysdera silvatica. So in order to perform all these comparative genomics studies or population genomics, first we need a reference genome. So last year we published the first draft genome of Dysdera silvatica in GigaScience. And this draft assembly was composed by 2 short-read libraries and 2 long-read libraries. By flow cytometry, we determined that the size of the genome was 1.7 gigabases, and the size of the assembly was finally 1.3 gigabases. However, this assembly was still a bit fragmented, and with the cheapest cost of the new technologies, sequencing technologies, we decided to improve it to a chromosome-level genome. So the aims of this project are, first of all, to improve the draft assembly of Dysdera silvatica to a chromosome-level genome, in second place, to annotate structure and functionality of the genome, and finally, to contribute to the study of the adaptive genomics and the trophic system of Canary and Dysderas. So first we send a Dysdera silvatica sample to the company Dovetail Genomics, and also we provide them our draft assembly, so they could sequence one Chicago library, another one HiC library, and using their HiRise scaffolding pipeline, they construct a new genome assembly. So once they provide us this assembly, first we polish it with the software Next Polish to correct contaminations or sequencing errors. We make the Busco pipeline to obtain completeness stats. And then we used repeat masker and repeat modeler to make an analysis of the repetitive regions and to obtain a submasked genome in which, in addition to RNA-seq data and a protein-related set, we use this as input to the breaker pipeline and obtain the structural annotation. Finally, we use BLASTP and interpro to search for functional annotations of the genes annotated here. So here we have the comparative between of the draft genome between the chromosome-level genome. The more remarkable things to say is, for example, that the total number of scaffolds were highly reduced from 65,000 to 15. Also, the number of genes annotated was reduced from 48 to 24,000, which is not a bad result because probably here there was the genes were more fragmented. And from these 24,000 genes, 90% of them have any functional annotation related. Another interesting statistic and important is that the N50, which shows the contiguity of the genome, have a huge increase from 38 kilobases to 174 megabases, which is very huge improve. Also, we have the results of the Busco-Araneae set. That Araneae set is a set of very conserved genes in arachnids that this pipeline searches in your genome. So the more genes it finds, the better. And we find an 80% of these genes complete in our chromosome-level genome. Another interesting data to say is that 80% of the genome was condensed in in the 7 larger scaffolds of the chromosome-level genome. And these 7 larger scaffolds are probably corresponding to the 7 chromosomes of Dysdera silvatica, being the larger one that is here highlighted, probably being the X chromosome having 317 megabases. All this information is corresponding to the karyotype experiments that are a personal communication from Mikel Arnedo's lab, that Dysdera silvatica have 6 pairs of autosomes and 2 sex chromosomes in females and 1 in males, as they have an X0 system. About the repetitive regions, the most important thing to say is that 52% of the genome is composed of repetitive elements, and most of these repetitive elements were classified as transposable elements. So to show something about the functional annotation, here we have the ionotropic receptors chemosensory family that is annotated across the 7 main scaffolds. And finally, to say the concluding remarks are that N50 and Busco analysis show a good contiguity and completeness of the genome. We annotated 24,000 genes, 90% had functional annotations. Also, we identified the 7 possible chromosomes at the assembly, and this chromosome-level assembly represents an important resource for future comparative and population genomic analysis. So finally, I want to thank to all my lab mates and Mikel Arnedo's labs, also to the organizers of this Congress, and to all of you for paying attention. Thank you very much.